All right, so we were synthesizing the aromatic amino acids in the last part of our lecture. The last move to create phenylalanine and tyrosine is transamination. And so we have an alpha keto acid at the top, and within a transamination, we take the amine from glutamate and we transfer it where the ketone was, and alpha ketoglutarate is the leaving group, and we have tyrosine and phenylalanine. All right, tyrosine is a complex reaction. What I really want you to understand about tyrosine is this corsmate is a intermediate that was in the other two amino acids. And so this is a branch point between the aromatics. The reaction here is quite complex and actually involves some enzymes themselves. And so I'm not going to have you responsible for this particular synthesis other than knowing that corsmate will synthesize tryptophan. Now, to continue on a couple of synthetic pathways, I need to introduce to you a methyl carrier. So ATP really it can be thought of as an ADP that's carrying around a phosphate that can be transferred by kinases. All right, and the electron carriers can be thought about as carrying a hydride around, like NADPH carries a hydride. There are a couple of organic compounds that carry a one carbon group. Now, it says a methyl carrier, and sometimes we will transfer one methyl group, but we will also transfer one carbon in other oxidation states. We'll see a formal group being carried, so a ketone with a hydrogen. And so we're going to introduce these methyl carriers. Adenosylmethionine is one, and that will be exclusively a methyl carrier. And then tetrahydrofolate, which is used to regenerate the adenosylmethionine, and then tetrahydrofolate will carry all sorts of one carbon groups with different oxidation states. Now, one thing about tetrahydrofolate, this is something you should probably know about any one of your professors. If they worked with something in their graduate studies, then that probably is something important to them, and sometimes it might make it on the exams. Dr. West actually studied the tetrahydrofolate enzymes when she was in her graduate studies. All right, here is adenosylmethionine, all right, and it is created in its beginning stages, okay, by reacting methionine with ATP. Now, if you take a look at the reaction for this, all right, usually when I ask you to talk about ATP synthesis and where I attack the ATP, there's something really strange in this, all right? Leaving group, all three phosphates. So what really happens is the electron pair becomes a nucleophile that attacks the carbon, the number 6 carbon, the number 5 carbon on the ribose ring, and all three phosphates fall off. Now, what you see is this very odd bond. What was the methyl on the methionine, and then now we have a sulfur in a positively charged state. Now, do sulfurs want to have a positive charged state? Not really. Okay, sulfur is relatively electronegative, kind of like oxygen, but then oxygen we do find in a positive state when we get the hydronium ion. Okay, so it can be that way. Yes?
Why do the three phosphates separate from each other?
Yeah, in actuality, this even gets broken down by inorganic phosphate to be 2 pi, so really they should have written 3 pi's coming. As it breaks down. The triple one will be broken down, and the double one will be broken down even further. It's a very strange reaction. And it gets a very strange leaving product. So this is highly not energetically stable. That we can talk about this activates that carbon. So if I attack that carbon, I have a wonderful leaving group. I can get rid of that positive charge. And what will happen is we can deprotonate an R group. That R group will attack at the methyl. And so if I take off the proton, this can attack at the methyl, and that creates a leaving group, S-adenosyl homocysteine. And we've transferred one methyl. Now this is kind of an oddball, okay? And what I'm going to get is homocysteine. Okay, one kind of question I will give you on the exam would be this. Classify that particular reaction. You have 5 options. 5 options are SN1, SN2, SN2, E1, E2, and E2 prime. Which kind of reaction is this one? SN2. The one thing that you do not see, which would be really indicative of an SN2, is inversion of stereochemistry, but there's no stereochemistry, so we can't see it. And so the adenosine, the actual, is that a nucleotide or a nucleoside? The adenosine. Well, I'm taking you back to high school now. Nucleotide or nucleoside? Oh. You'd be guessing completely, right? All right. A nucleotide is a base, a sugar, and a phosphate. A nucleoside is a base and a sugar. And so this would be the nucleoside, the adenosine itself. All right. And then we've got homocysteine. Classify homocysteine for me. What kind of amino? It's an amino acid. Sorry, I answered my own question. All right, it's not one of the 20, but it is an amino acid. All right, here's tetrahydrofolate. All right, look at how complicated that is. You will see this a lot, THF. Okay, because it is such a large cofactor. Now, THF, you may have heard about this in organic chemistry. Do you know what THF is in organic chemistry? It's a solvent. It's called tetrahydrofolate. And so this is where science acronyms get really, really messy. In one case, it means something totally different. All right. We're going to focus in on the ring structure, and we're going to focus in on the 2 nitrogens. Those 2 nitrogens are able to carry the carbon units that we will be transferring. And so here I only show part of the THF. We're going to have a methyl on the 5th carbon, the 5th nitrogen. And so the homocysteine will react. Okay? In that case, if I articulate the hydrogen, a base will take the hydrogen, the sulfur will be negative. I will attack at the methyl group, and that will pick up the proton. And I get methionine back. And so it's a circular pattern. All right, we're going to use that methyl in a little bit to complete a reaction. All right, and so methionine plus ATP gets the adenosylmethionine. That's the one that can transfer the methyl group. The adenosyl homocysteine is reacted to let the homocysteine go back out, and then the methyl group is transferred from THF. The adenosine gets phosphorylated back up into ATP and reused that way. We've looked at glutathione before. This is also a nitrogen-containing group. All right. It looks like a peptide. It is a peptide. But what is strange about this peptide? If you have an order of peptides, what goes at the beginning and what goes at the end if you have a string of peptides? Which terminus and which terminus? What's at the end terminus? Not a nitrogen. It's the carboxy at the end. At the beginning is an amino terminus for all proteins, for all peptides. All right, that's a peptide. What is weird about glutathione? You have two carboxy groups. All right. You also have an N-terminus and a C-terminus. How did I end up with two C-terminal, or two C-terminus, one at one end and one at the other? What is strange about the peptide bonds here? Yeah, the first one, glutamate's backwards. It makes a peptide bond with the R group's acid. Normal peptides are made with the acid on the alpha carbon. And that's why it's the gamma glutamate. Okay? So it's a short peptide not made by the ribosome. This is not encoded in DNA. It is actually created by 3 separate enzymes to make this glutathione. What is important is this sulfur. All right? This sulfur has a hydride, and this is basically a hydride carrier. Okay? And it actually is very concentrated in the cell, one of the most concentrated molecules in the cell. When it reacts, 2 of them can come together, and it can give 1 proton and 1 hydride as a leaving group. This hydride is normally used to react peroxides, and it is used to prevent oxidative damage. It will use an enzyme called glutathione peroxidase, and what happens is it's going to use 2 of these glutathiones. The first glutathione gives its electrons to the selenium atom inside the enzyme, and the selenium atom is going to react with a peroxide to create a hydroxide, which is not reactive. This is very reactive. And then it creates an acid. A second glutathione will come in and kick off water, and it will be interacted with the sulfur, and then this one, to finish the reaction out, this sulfur and that sulfur get together to have the oxidized version of glutathione come off in the reaction. And so this is synthesized to have a very large concentration of hydrides used to protect against oxidative damage. So that is also a nitrogen-containing compound that's important. Heme synthesis. Okay? Heme synthesis is important along with its degradation. And what we will use is 2 molecules to start the synthesis. Glycine, an amino acid, and succinyl-CoA from the citric acid cycle. And the reaction actually occurs in the mitochondria, and I make an intermediate that is transported into the cytosol. All right? And then we react 2 of those to create this ring structure, and we take 4 of those to begin to make the heme group, of which I've got to add an iron in here later. All right? This reaction is kind of interesting or important, and you can see that I'm going to make a ring structure with a 5-membered ring, and the reaction occurs with 2 of these. All right? And so if I actually do some inventory for this, okay, I've got an acid up there. I've got a CH2, a CH2 in the yellow here, and then all of a sudden this carbon right here, okay, is different than it was, because that was a ketone. Okay, if I do 1, 2, 3, 4, 1, 2, 3, 4, that's the ketone, so 1, 2, 3, 4, that's the ketone. All right, I now have 1 methylene left on this, And then I have the ammonium. Okay? The reaction can be facilitated this way. You've got to connect the 1, 2, 3, 4th carbon of this one, to the 1, 2, 3rd there, this. So this one and this one has to come together. Then I look over here, 1, 2, 3, 4 has to be reacted with the nitrogen. So this one has to be reacted with this one. All right. Two good things. I've got to make the blues come together, and I've got to make the reds come together. Now, when you do these reactions, you got to decide which one goes first. Okay? Would I react the blue two entities together, or would I react the reds together? And would you see why, other than just guessing red or blue? Yeah. It doesn't matter. It isn't, because that's if you actually look at that, they are not left and right. That is an sp2 hybridized, so you are trigonal planar, and so you would have to draw that in a triangle shaped, and it can rotate each way. So yeah, this is where a flat piece of paper really kind of, you lose the three-dimensional structure, so it doesn't matter which way you draw that. All right. How do I attack ketones? What have we seen that attacks ketones? What does the atom need to be to attack a ketone? What are the properties of a ketone that you know? This is one of the big picture things I want you to understand. There's a ketone. All right, we've attacked ketones, but why? All right, I have two atoms, carbon and an oxygen. What are the differences between those two atoms? There's a C and an O. Oxygen has a lot more electronegativity. That means there's a partial negative here and a partial positive there. So the carbon is partially positive, which then, if I'm going to attack something there, what charge does it need to be to attack? Negative, okay? And so, how in the world, or I have to have a lone pair of electrons at least, all right? So, nitrogen has some protons, and they can be deprotonated, right? And because the nitrogen will have to attack the carbon itself with a methylene it isn't acidic. But tell me something about the methylene on the blue. This is another principle that I wanted you to get as we look through everything. Ew. Booger. Lost it all. Okay. The other principle was this. If I have a proton like this, okay, then these protons right here are acidic. Okay. If by chance I take one of these off, you would have a negative charge on the carbon, which is not great. But I have a resonance structure that allows that negative charge to be there. And so these would be actually acidic. But they're only acidic if you have an acid there. Okay? And so if you reacted these two things, this was blue, that was blue, this was red, that was red, you would have to do one before the other because you would lose the fact why one was acidic. Okay? If you attacked this here to create the alcohol, which would be the first step in the mechanism, then the blue methylene would not be acidic anymore. If you actually deprotonate this one and allow it to attack here, and that would become an alcohol in the middle of the reaction. This is acidic. This still can be deprotonated, and then the red becomes the second attack. If you do the red first, there's no organic reason why the blue would react. If the blue reacts first, the red can still react completely. And so mechanistically, you would have to have the blue make the bond first to create this bond first before you make this bond. So this one becomes first, that one becomes second. Also, there are eliminations that have to occur when those hydroxides are made. You can eliminate it to create two double bonds. Can you tell me why we have that ring structure and we would eliminate and it is really, really stable, that particular ring structure? Is there any organic reason why that ring structure is really stable? Because of, yes, but it's a special type of resonance. You should be able to see this. It is a resonance. That's right, the electrons on top of the nitrogen ring. Okay, and what would happen if that moved there, then that moves there, then this moves here, and it spins around completely all the time. What is that principle? It is, you have something that is very similar in organic chemistry that spins around the electron pairs. What is the principle or the stability here for this ring structure? Yes, thank you, it's aromatic. And so that aromatic is very, very stable, which drives the reaction, okay? And so, somebody knew it, wow, I'm so pleased. Okay, all right. Heme is important because it's in the blood. That's good. And what does it do in blood? It carries oxygen in hemoglobin, okay? Now, one thing that is always done when you do blood work is you look at what's called the bilirubin concentration. And bilirubin is this structure right here, okay? And if it is high in concentration, your skin becomes yellow and you're jaundiced. A lot of babies have this particular issue. We use light to treat that for babies, okay? Why would light be a treatment to degrade bilirubin? What's unique about the structure that you see in bilirubin? And something about light. It was also found in the structure of vitamin A, which was part of the retinal that was in rhodopsin. It's also part of the system in chlorophyll that it's important. What in the organic structure of bilirubin allows light to be a treatment? what you see in all of those organic molecules, the ones that are in the photosystems and the retinal, is their chromophores, is the double bonds that are conjugated. Conjugated double bonds allows light to be absorbed. That's the organic structure that allows light to be absorbed. And if you absorb light, you put them in excited states, and you tend to allow them to react and be degraded. We also look at bilirubin's content to see in blood work to see if your red blood cells are being broken down. And so the breakdown of red blood cells can indicate anemia and other problems that could be like kidney problems and things of that nature. And so bilirubin is something that is in some of the basic, just general blood tests. As you start to break this all down, they become yellow colored and brown colored. And so the idea of the coloring that's coming out in your waste products are from these particular molecules themselves. And so a lot of people think urea is what causes urine to be yellow. No, that is not true. It is actually breakdown products of the heme. And in the brown color is for your feces. All right. So amino acids were very, very important nitrogen-containing compounds, but more or just as important are the nucleotides. You can almost think of them maybe even more important because not only are they part of the genetic material, but they're also derivatives of activated intermediates. You've got the energy transmission. We've seen ATP, GTP, UTP used as energy. All of the cofactors, electron carriers, and the activated carrier for degradation, CoA, is part and has a nucleotide as part of its structure. Also regulatory molecules, including cyclic AMP. So nucleotides are everywhere, and so synthesis is important. In this, a molecule called PRPP is a very important molecule in terms of both or all of the ring structures, and it is the ribose ring that begins the monophosphate, diphosphate, and triphosphates. In this, what we are going to have is one attack that you rarely see. You will take and deprotonate the hydroxide because that is the only change in the reactant to the product. This pair of electrons attack at the alpha, beta, or the gamma. It's at the beta. This is very rare that happens. Kick up, kick down, and AMP is the leaving group. And we get ribosyl 1 pyrophosphate. This is not only used for the synthesis of the nucleotides, but this structure is also used for a couple other intermediates, including amino acids. So this is a branch point that's very important. Where do we get the ribose from? What pathway? Pentose phosphate pathway, very good. And it's at the cost of what structure coming in from your food? Where do you get the ribose from? The easiest is, what kind of foods do you eat? There are 3 kinds. Sugar, fats, and proteins. So which is the easiest to get from? What starts the pentose phosphate pathway? Glucose. Yes, so carbohydrates are the easiest ones to get it. You can actually get glucose from amino acids, although it takes the amino acids into the citric acid cycle up through gluconeogenesis. You cannot get ribose or glucose from fat. And so when you think about how metabolism is integrated, I can get the ribose from amino acids, or I can get ribose from carbohydrates. Carbohydrates are the easiest. All right, star this slide. You need to know this particular one. where all of the atoms come in from the purine synthesis. Now, what's interesting about this synthesis is that almost all of them come from single atoms come from large components that bring them in. And so the nitrogen here comes from glutamine. The tetrahydrofolate is going to bring this carbon in. CO2. If you see in another book bicarbonate, that's exactly the same thing. It's just dissolved, reacted CO2 with water. And so a lot of times that will be bicarbonate or CO2. Aspartate, THF, glutamine. The largest component, these three atoms, are from glycine. And so lots of things come in to donate individual atoms. All right. We start off with PRPP in the structure, and what we're really going to do is build the base pair onto the ring structure. To do that, glutamine comes in, and we react with the R chain. The base will be deprotonated, or will deprotonate the water. The hydroxide will attack at the carbonyl. Electrons will kick up, electrons will kick down, and an amine will be given off. Okay. And the rest of the reaction proceeds to create the base pair, or the beginnings of the base pair, the amine on the sugar. And so if you flip back, it says the amine, that nitrogen, comes from glutamine, and here it is. The red arrows represent what kind of mechanism of the five that you know? SN1 tends to have a carbocation in it. SN1 in biological systems tends to have a retention of stereochemistry. The pyrophosphate on the bottom is in the alpha or beta conformation when we talk about a ring structure. Help you out? Alpha. The nitrogen at the end is in the beta. This is an absolutely classical SN2. We see inversion of stereochemistry at the center that was attacked. If you take a look at all base pairs that are on ribose rings, they're always in the beta conformation. All right, the next reaction, okay? What we're going to do is we're going to react an entity to add on to the ring. This is an amino acid. What amino acid is this one? Can you recognize it by the structure? Yes, good. Other than that, other than the fact that just a couple slides ago it said glycine was reacted, that's a good part of it too. All right, the glycine will react, and if I take a look at the reactant, the two reactants and the product, it seems that no change, the ketone, no change. The oxygen now has a phosphate to it, and so the oxygen must attack. I do not have to deprotonate because it's already negatively charged. Will I attack at the alpha, beta, or the gamma? The gamma, because I transfer only one, and ADP is a leaving group. And I have a activated glycine. All right, activated means around the ketone. In this particular case, I have a wonderful leaving group, a phosphate that leaves. And so if I look at the two reactants now, the glycine and this sugar, I can see that I have to put this nitrogen, which is right here. It has only one hydrogen. Here it has two. And it has to be connected to the ketone right there. Base. We'll take that. We'll attack. I will kick up. I will kick down. And I will kick out. And I have now a ring structure that has 4 members of the ring. So that actually did a lot in the reaction there in terms of forming this ring. All right. We have THF coming in. Before, when we reacted with the adenosylmethionine, we actually had the methyl group on the 5 nitrogen. Here we're carrying the carbon that's in the form of a formal group on the 10th nitrogen. And so I am bringing in 1 carbon from tetrahydrofolate, and if I take a look at the reactant nitrogen in the product, I have 1 hydrogen, I have 2, so I must take one of those off, and then it has to be attached to a ketone, and there's the ketone. For clarity, I will take that proton, although I could take either one because they're equivalent. I can attack, kick up, kick down, and in this case, this will pick up a proton. All right, I am beginning to make a ring. All right, this reaction's kind of strange. We're going to see it a couple times. I'm not going to give you this particular reaction on the exam. A little bit too complex, but glutamine plus water gives the amine that comes off was the first reaction that we had. This happens a lot where the glutamine's R group is attacked by water and an amine becomes the attacking group. So that is a principle that you can use quite often, but we're going to have the amine attack at this particular carbon. You'll get a hydroxide intermediate. It will then react with the ATP to make a good leaving group, and then I will eliminate that. This type of reaction right here might be on the exam though, and it would be an exam type of question could be from here to this alone. So the exam question could be from here to here. All right. In this case, you can see we are going to close the ring. So there's a ring structure here, okay? And so that means something has to connect, and so what connects is this nitrogen has no hydrogens, this nitrogen has a hydrogen. I've got to get rid of that. This nitrogen is now connected to a carbon right here that must convert into a hydroxide. The only carbon that can convert into a hydroxide is this one right there. So I take this, attack that, and I pick up a proton. All right. Next reaction, if I go from the reactant to the product, okay, I look around the ring, no change, no change, no change, definitely a change. That is gone. I have a double bond. Nitrogen has no hydrogen. The way I wrote this mechanism, if you had to classify that reaction of the 5 that you know, what would be this one as written? SN2? The 2 is correct. Did I substitute something? Was something there and then I substituted on the same carbon? No. I've done what? Elimination. So this is an E2. Okay. That will also be a type of question where I'll draw out the mechanism, have the product, and you'll have to identify. All right. Interestingly enough, if I look at this and that, all right, this is an isomerization. Because if you actually add up all the hydrogens, nitrogens, carbons, phosphoruses, nothing has changed in terms of the numbers of things, but I've rearranged. All right. The rearrangement happens if you take a look. How many hydrogens are at this carbon here? 1. Here I have 2. And here is 0, that's still 0. Here is 2, that is 1. And there I get an isomer. The isomer that is at the end product is very stable because it is another ring that is aromatic. And that's what the driving force is to close that ring. All right, the one thing about it, purines are a double ring, and so I do not have one of the standard nucleotides that you would recognize. And so I've got to continue making the ring, and the next ring is going to be, I believe, a 6-membered ring. And so bicarbonate reacting with ATP will create an intermediate that has the bicarbonate ion that has a phosphate. So that makes this carbon activated, so something attacks because this is a wonderful leaving group. And I can see between the reactant and the product 1 hydrogen 2. I can deprotonate one of those. The electron pair will attack. Up, down, the phosphate is out, and I have a carbon. Now, if you go back to the slide that says where all of the atoms come from, this says a bicarbonate ion, but in that ring structure it said it came from CO2. Remember, bicarbonate is an equivalent of CO2 that has been dissolved and reacted with water. And so, depending upon what book or depending upon what standardized exam, that could be one or the other. So if you just memorize CO2 and then you see bicarbonate, it could catch you off guard. This is a really complicated reaction. It has a shift that occurs. This would not be a reaction that I would ask of you. This actually becomes covalently linked to the enzyme as part of its reaction. So this would not be one that I would ask of you. All right, this one though could be in terms of the reactions. And so I've got this ring structure that I'm trying to create around. I've got this acid sitting here, okay? But in the product, okay, the acid actually now has a phosphate. Okay, I will attack this. Okay, and I will get the phosphorylated intermediate. I did not name the enzyme that created this particular product. Oh, and I'm over time. But what kind of enzyme would that be? A kinase. And so